Hey everybody, today we are looking at section 8.3, which is cell transport. So this is going to tie in with really well with the stuff that we ended section 8.2 with, which dealt with cell membranes. And you can see here in this image we have a phospholipid bilayer. This is my cell membrane, the exterior of that cell out here, and then some different molecules kind of floating around in that space and additional cells back there. So what we're going to deal with here is exactly how we move materials into a cell, how we move materials out of a cell, and what kind of drives that process, whether we need some energy to help with that, additional proteins or other molecules or organelles. Okay, so go ahead and get the title, section 8.3, cell transport. All right, so learning objectives for this section. Oddly enough, these are probably the two shortest learning objectives that we'll see maybe all year from this textbook. Explain how passive transport works and explain how active transport works. That's it. And I think just looking at that vocabulary that you're seeing right here, passive and active, that should give you a good indication about what these will entail, right? Passive meaning if something's super passive, it doesn't require usually a lot of energy. And active is kind of the opposite of that. And that's exactly what we're going to see here with the types of transport we're going to look at here today. Okay. So we're going to start with passive transport, which is the simpler of the two processes. As it states there at the top, it does not require any energy from the cell. So I usually use the example of students who like to sit in my class kind of passively, like those who don't engage much, those that don't shout out or disrupt. So you can kind of take it in both ways, but either way, they're not exerting a lot of energy in that case, right? They're maybe even sleeping in class, and we all know people that have had a habit of doing that sometimes. So passive transport, again, no energy required. What it does require, however, is a concentration gradient, which refers to this idea that we have a difference in concentration in one place and another. And when we talk about concentration in this section, what we're going to really refer to is our solute concentration. Okay, that's the one that we're talking about with this. So in other words, the amount of solutes dissolved in a solution of some sort. And usually in a cell, a solute looks like either sugars, salt, or sodium or chloride ions. It could be proteins and lipids and other things, but these are the molecules that are floating around in our cells that can change in concentration based on what's happening across the cell membrane. Okay, what we know happens in passive transport, the reason why this occurs is kind of and does not require energy, is based on this idea that there's a difference in concentration of those solutes. So in other words, this is like the natural motion of particles trying to kind of balance themselves out. Okay. So we move down a concentration gradient, which is saying we go from a high concentration with a lot of particles to a concentration where there are very little particles. So an example of that would be something like this. You've got a whole bunch of little particles, and maybe on this side of the room, I just have 3 or 4. Okay. And so what we know happens is these guys are awfully kind of contained in this space. Then they're not going to want to kind of sit there all together. They're going to want to be able to kind of spread out. And as they do that, the natural motion is going to kind of take them in this direction so that we end up with a scenario where they're all kind of equally spaced out. We know this happens in gases all the time, right? Gases will move and the particles within them move to kind of fill up a space. And really, it's kind of gross to talk about, but one of the examples we always talk about with this section is farts. Sounds weird, but we all kind of have an understanding of how a fart works, right? I mean, when you have to pass gas, the initial concentration, the highest concentration, is going to be right at that person that had to release that gas. But over time, those molecules don't just stay in that space, right? They're going to spread out, and eventually sometimes other people can smell things and it gets kind of gross and weird, but that's that idea. We are diffusing things. Same idea with air freshener, right? You spray air freshener, the highest concentration of that air freshener is right where you first sprayed it, but that diffuses, right? The whole idea is that it diffuses in the room, it passively gets transported through the room, and it goes from this high concentration to a low concentration. So I use the word diffuse a couple times. That is an example of passive transport. It's one type of passive transport, and it involves, again, simply molecules moving from high to low concentration. There is also facilitated diffusion. We will come back to that in a little bit. But osmosis is one we're going to spend a lot of time talking about today, and we'll get to that here on the next slide. But that is specific to water. So osmosis is another type of diffusion, which is a type of passive transport. And osmosis looks at specifically the movement of water from a high concentration to a low concentration. And so if we look at this process of passive transport, let me see if I can, let me go back. So when we look at passive transport as it's occurring, what tends to happen across the cell membrane is something that we see, like for example, right here. So we have our cell is in this case right here. We've got the interior of the cell here in yellow, exterior of the cell here is in blue, and our solutes are represented by these little blue spheres. Okay, so if we have a scenario like this would be step number 1 in this case, right? This is where we're starting. This is our starting point. If we have a scenario with a lot of solute on the exterior of the cell, what tends to happen is as long as that solute is small enough, hydrophobic enough, nonpolar enough, and uncharged enough, it's going to be able to kind of start crossing this membrane. And we know that those molecules are going to try to kind of reach that equilibrium to kind of make themselves the most comfortable in their space. And so the higher concentration of that is going to drive the movement of these molecules on the outside to the interior of the cell. So we get a net movement of solute particles from the side of the membrane that has a higher solute concentration to the side that has the lower. Okay, now there will be some molecules like you see here with this arrow that will still go the opposite direction because these particles, like in everything, are moving constantly, and sometimes that movement may take them back out of the cell. But in the second stage, the majority of the movement, as you see, is going to be into the cell or down that concentration gradient from high concentration on the outside to low concentration on the inside. And I could add that here. So we started with high on the outside and low on the inside. All right. So as this continues, all right, and these particles will keep moving the entire time again, they will eventually reach what we call equilibrium, which is where our concentration values are exactly the same. In that case, particles will still continue to move. This is panel 3. And notice now there's no like net gain or loss of concentration in this case. The concentrations will remain equal. The number that come in will equal the number that leave, and so on. And so we have these additional now arrows showing that we're pointing the opposite direction. So molecules still move even when they're at equilibrium. It's just there's no net gain or loss of these across the membrane. Okay. So moving on to look at specifically diffusion and then also osmosis. So diffusion, again, like I mentioned, is a form of passive transport. It's moving from high to low. I mentioned the air freshener example and the fart molecules example. We're looking at something very similar here on this screen, which is this example of adding food coloring to water. And if you've ever done that before, you know that what tends to happen is the food coloring molecules, those colors that you see right here, aren't going to remain in this one tiny little space, right? They want to diffuse, they want to spread out in this, especially if you give it a stir and kind of mix it up. And then they don't separate out after that, right? They mix through that whole liquid. They turn the whole liquid, in this case, probably pink and blue and green. And that liquid will stay that color because we formed a homogeneous mixture, a solution. So when we talk about diffusion, regular old diffusion, when I talked about air freshener and farts, that is an example of what we call simple diffusion. You do not need cells or living things really to do that. All right. However, diffusion can also occur across the cell membrane in living things. All right. And so that will be the example that we look at specifically with something like water right here. This example on the right is actually something called facilitated diffusion, which is another form. And facilitated diffusion, by definition, requires the use of cells, cell membranes, and then transport proteins in the membranes. And the reason why we need transport proteins here is because the molecules that are trying to cross the membrane have those properties that kind of contradict with what we see in the membrane. So what I mean by that is they may be large molecules, they may be polar molecules, they may be charged molecules, and they may be hydrophilic. All right. If any molecules have any of these properties, even if they're going to go from high to low concentration, even if they go down that concentration gradient like we saw, it's still going to need the help of these helper proteins. These are essentially channels in the membrane that provide a passageway for molecules to come through. So the example here that we're looking at on this picture is of water. Water obviously is a polar molecule. That's something we talked about early on in chapter 2. And as it's a polar molecule, it can't interact with these fatty acid tails that make up the inside of this membrane. But we do know that water has to get into cells. And so we use these specialized protein channels called aquaporins that provide a space, a passageway specific for water to move through into a cell or out of a cell. Okay. So facilitated diffusion, I can't emphasize enough. We are not using energy for this. We still go from high to low concentration or down a concentration gradient. However, we need the help of a protein to provide a passageway. So there's nothing really that a cell is actively doing in this process. It's simply providing that like little hallway or that little channel or canal that allows these things to get through. Okay. So that is two different types of diffusion. So regular diffusion or simple diffusion and facilitated. Now remember, I also already mentioned that osmosis is a form of diffusion. And here is our definition for that. So this is the diffusion of water across the semi-permeable membrane, like the cell membrane. As with all forms of diffusion, the concentration of my solutes in my solution will determine how these things move. We still go from a high concentration of water to a low concentration of water. However, there's something that really gets tricky when we look at osmosis, which is that scientists, for whatever reason, when we first started talking about osmosis, decided to give special names to identify solute concentrations and not concentrations of water, which is kind of counterintuitive. We're looking at here movement of water, but we're going to spend more time here talking about solute concentrations. And remember, water is always a solvent, not a solute. So when we look at a solute concentration in a solution, including water as a solvent, we know there are a couple of really important things we can keep in mind. In general, the more water you have, the more diluted the solution is, the lower the number of solutes. Like think about if you've ever made sweet tea or Kool-Aid and you've added maybe too much water. It probably doesn't taste very good, and it also probably doesn't taste as sweet as it would as if you'd added the right amount of water, right? Because you've diluted the concentration of solutes. You decreased or diluted the concentration in the amount of the flavoring and the sugar by adding too much water. However, on the opposite side of that, the statement at the bottom, if you make Kool-Aid or you make sweet tea and you don't add enough water, then you're going to get a very sweet, very concentrated in terms of flavor solution that's going to have a high concentration of solutes. And so this is kind of what I want you to try and keep in mind as we work through these things, is thinking about, okay, if I have an area inside or outside the cell with a low amount of solutes compared to something else, that area with a low amount of solutes is actually going to have the highest amount of water. And then vice versa. If I have an area with a very high amount of solutes, well, that in comparison to another area, whether it's in or outside of the cell, if I have a high solute concentration, chances are I have a very small amount of water. Okay, so this is kind of where we're headed with this osmosis stuff, and this is where things get kind of confusing. So the picture that you're looking at here at the bottom is this U-shaped tube, okay? And we talk about this in biology a lot because this is a really good way of kind of representing how osmosis occurs, how it can occur in living things. And so in this U-shaped tube, notice we have a couple of things going on. We have solutes that are represented by my dots, and those are sugar in this case. I have a high concentration of sugar on this side, and we can go through and count. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 sugar molecules on this side. And on this side, I have 1, 2, 3, 4, 5. And that's it. So if we think about water volume as being exact same, which we can tell in this diagram it is, if I have 11 sugar molecules here and only 5 on the other side, I have a difference in concentration for both my water and my sugar molecules. Now, sugar is something that is polar, so normally in a cell membrane, it's not going to cross on its own. Water has the exception of having those cool little aquaporin channels that allow it through. So if we equate this area down here to like a cell membrane, well, we know that water can get through, but the sugar cannot. All right. And as a result of that, if sugars are kind of stuck where they are, water's going to move in this direction from the left to the right to try and dilute that concentration there of the sugar on the right. And so what we see is the volume increases over here, the volume decreases on this original side, and that's to kind of try and equal out the concentrations. And if we look at what kind of how those molecules are spaced out in this diagram on the right, you can see those concentrations are probably pretty similar at this point. So the water level, we're actually adding water in this case to lower the sugar concentration. This is the same kind of process that happens in our cells when we look at imbalances in solutes, whether it's outside of the cell or inside of the cell. We're always going to look at specifically moving water as a result, and movement of that water from the high concentration of water to the lowest concentration of water, or said differently, from the lowest concentration of solutes to the highest concentration of solutes. And I know that gets tricky, okay, when we start talking like this, but you kind of want to keep all of that in mind as we go through this. So when we look at osmosis as it affects specifically living things, we generally like to use the example of things like red blood cells, which is the image that you look at here. There are 3 different types of solutions that we have kind of given names to describe the solute concentrations found in those locations and to talk about them kind of in a meaningful way. So the first one, the easiest, is to say that we have an isotonic solution. And if you remember from Latin roots, iso means equal to. So if I have an isotonic area, whether it's inside of a cell or outside of a cell, that means that my concentration of solutes is equal to the other thing, whatever it is I'm comparing it to. So that means if I have, in this case, let's say I have 1, 2, 3 solutes. Those are really terrible solutes in this case. Let's say I have 3 solutes in that cell, then I would also have the same number out here, but they would be spaced out kind of as in an equal distance from one another, right? But that's per unit of water, right? So isotonic, what tends to happen in that case, and you can see from the arrows, we still get water movement because, again, molecules are constantly moving in these scenarios, but there's going to be no net gain or net loss of water in this case. So isotonic conditions like this is what our cells thrive the best in. It's where we like to be in terms of animals and survival. We want our cells to be isotonic with their environment or with the solution in which they're sitting. So all that exterior space surrounding a cell in our bodies, we want those to always be isotonic, okay? And so the movement of water kind of drives that. So this is usually where we kind of try to end up following osmosis. This tends to be like our final product. But again, water is still moving in that case. Now, we do have scenarios where cell shape will change as a result of water movement. And one of those is what we're seeing right here. So the term that you've got here is hypertonic. Hyper, if you remember, means over and above. And when we're describing a solution as being hypertonic, that means that we have higher amounts of solutes in that solution as compared to something else. So in this example that you see here in this image, I've got a hypertonic exterior environment, meaning I have a lot of solutes out here, like a whole bunch of solutes out here in this space, but inside my cell, I probably just have a couple. Like it's not nearly equivalent, the outside and the inside of the cell. I've got a lot more out here and not as much on the inside. And so what I like to think of with this is that this would be equivalent to having a lot of pressure coming on a cell from the outside. Like they can feel all those solutes and they can feel that pressure. And as a result of that, they want to do something to fix how uncomfortable they are. The only option that a cell has, because we're going to assume, again, our solutes cannot move, in an osmosis, they don't move. Water is strictly really the only thing that's moving in these scenarios. When that happens, water is going to be kind of sent out of the cell, and water sent out again to try and lower the concentration of solutes on the outside of that cell. So that pressure that that cell is feeling, if the cell releases some water and dilutes that concentration, that pressure will also decrease a little bit. So cells that are put into a hypertonic solution, whether it's purposeful or not, will always shrink in size. Okay, cells in a hypertonic solution will shrink in size because they'll lose water to the exterior environment to try and dilute or decrease that solute concentration. Alternatively to that, we have the opposite scenario here. If we take a cell and we put it into a solution that we call hypotonic, well, hypo means under or below, and in this case, I have very few solutes on the outside. Let's say I've just got like 3 out here, and I've got a whole bunch of these little specks of my solutes here on the interior, okay? There's all sorts of them. In this case, the cell's going to feel lots of pressure on the inside, and as a result of that, again, if water is the only thing that's moving, the only option is for it to absorb water and pull it in. And then in that case, what ends up happening as a result of that is the cells can actually swell up in size, kind of like a water balloon would swell up with water if you were to fill it up. So water actually goes into a hypertonic cell if that cell is placed into a hypotonic environment. A couple of interesting points to make about this. The terms hyper and hypo are comparison terms, and I didn't mention that yet. But notice I have both on this screen and I had both on the last slide. What we're looking at with that and what we mean by that is these are terms that are equivalent to saying bigger or smaller or taller or shorter. And so if you think about the meaning of those words, well, we all kind of understand that it doesn't make sense if I just say Miss A is taller, because your immediate response is going to be than who, right? Or taller than what? Because you have to use a comparison word. So when I say a cell is hypertonic, well, it's hypertonic in comparison to something. It's not hypertonic to everything on the planet. It is just usually going to be hypertonic to whatever it's exposed to. So it may be hypo, it may be iso. But if you have the word hyper, you're going to have hypo in the alternative environment, okay? So regardless of which one is given to you, assume that the opposite is true of whatever it's being compared to. And of course, if it's isotonic, that's like saying student A and student B are the same height, right? You don't have to use that word more than once to imply that they are the exact same and they're equal. And that's the same that you get here. If something's isotonic, then the other is also isotonic. Okay. Also, another point to kind of make real quick. I don't know if you noticed this, but water in every scenario that we're looking at here is always going to go to the hypertonic space, okay? Because we're always going to try and dilute the higher concentration. So in this case, water went into the cell because the cell was hypertonic. If I scoot back one slide, notice water here went out of the cell because the solution here was hypertonic. So one of the general rules of thumb I talk about in class is to say, water always goes to whatever's hyper, okay? And that may be an important thing that you could jot down here too. All right, so a couple examples, some of the things that we just talked about to kind of review those. These different terms. Isotonic, again, means we're going to be moving water into and out of a cell at the same speed. In an animal cell, this is the ideal condition to have an isotonic cell in an isotonic environment. That is what our red blood cells should look like. Plant cells, however, that's not the best scenario for them because plant cells need to be able to do things like stand upright and hold lots of water in their vacuoles. All right, so it's not exactly the best scenario. They'll kind of slump over if they're actually exposed to this isotonic condition. If something's hypertonic, right, water is going to go out of the cell in both scenarios, and this is certainly where plants run into even more trouble, right? If they're lacking water, they kind of start to lean over and may actually bend and break if they have softer stems. And in that case, that's not a great scenario. In red blood cells, our red blood cells will shrink, and that can also be problematic. Hypotonic, on the other hand, this is actually the ideal scenario for a plant cell because it absorbs the most water, it keeps it within the cell, and it helps it to kind of enlarge its vacuole and stand upright and push out on that cell wall. So that's actually where plants like to maintain their cells. Hypotonic for us can be problematic because if we take in too much water in that type of environment, cells can actually lyse or burst or explode in the body, and that would be a problem, certainly. Okay. So our cells in our bodies like to maintain and remain isotonic, all right? And we do that again through those osmosis processes. All right, last two slides deal with something called active transport. And active transport is what you would hopefully think of it as being, which is active. And if something's active, that means we are using energy. Energy in the cell that we're going to be using is in the form of this molecule here called ATP. And ATP is something we'll talk about and come back to in chapter 9. But ATP is required. We also need the help of transport proteins or vesicles in these processes. All right, so there are a couple different types of active transport. We'll look at them on the next slide. But what you see right here, this first one is called a protein pump, and this is active. You can see ATP used right here. These things are actively going to move molecules against a concentration gradient. So instead of going down from high to low, active transport is going to move molecules from low to high. So it's kind of artificially pushing them in a direction that they don't naturally want to go, which is why it's going to require some energy. Okay, so protein pumping, that can happen with all sorts of different ions and molecules in cells when a cell is trying to create things like sometimes a concentration gradient. We have endocytosis here and exocytosis, which are two things we're going to look at here on the next slide. But notice we've got our Latin roots right here, endo meaning inside. This is a movement of molecules into a cell. And exo meaning out or outside. This is the movement of things out of a cell. Both of those processes require the use of vesicles. All right, so both of these require, vesicles. That V looks kind of weird. Oh, and the E looks even worse. So vesicles are used in this process. And those vesicle organelles, if you remember their function, is to move or transport materials within or throughout a cell. And so as part of this process, if we go to take in something through endocytosis into a cell, we have to require the use of vesicles to pull those things in. The way that it works is the cell membrane kind of reaches out and grasps whatever it is and brings it in, and that piece of the membrane that was kind of interacting with that molecule that was brought in actually becomes the vesicle itself. There's two different types of endocytosis. There's phagocytosis, and if you remember, phago means to eat, and also pinocytosis, which is to take in liquid or cell drinking, okay? Both, again, require ATP, both require vesicles. Last item here, exocytosis, is a process of removing or pushing out materials that are no longer needed. This can happen after the lysosomes absorb things, chop them up, and then send them on their way. Or after the smooth ER does detoxification, it can send it on its way. Again, you need the help of vesicles to do this. Those actually move towards the membrane, and then they kind of fuse with the membrane and push all their contents out, okay? All right, so that covers all of section 8.3. If you have questions, as always, let me know. Otherwise, make sure you get the rest of your work done, and I will talk to you later. Bye, y'all.